Revolutionary plant virus, a new hope in cancer immunotherapy. Imagine a tiny virus, known for infecting humble black-eyed peas, now holds the key to unlocking a powerful, affordable cancer treatment. This discovery is nothing short of revolutionary, but as we'll see, it's also a tale filled with twists and turns that challenge our understanding of how the human body fights cancer. Plant viruses might sound like a minor player in the world of medicine, but researchers at the University of California, San Diego have stumbled upon a game changer. They've discovered that the cowpea mosaic virus, CPMV, a virus that typically infects legumes, has a unique ability to trigger our immune system to target and destroy cancer cells. But here's where it gets controversial. While many viruses can wreak havoc on our bodies, CPMV is a rare exception that seems to awaken our immune system to fight cancer. This isn't just a simple case of a virus causing harm. It's a complex dance of biology that could revolutionize cancer treatment. And this is the part most people miss. CPMV doesn't just trigger a fleeting response. It reprograms our immune system to remember cancer cells, paving the way for long-term protection against the disease. In a groundbreaking study published in Cell Biomaterials, researchers led by chemical and nanoengineers at UC San Diego delved into the mysteries of CPMV. They found that this virus activates a wide range of immune cells, including neutrophils, macrophages, and natural killer cells, to infiltrate the tumor microenvironment and destroy cancer cells. But that's not all. CPMV also activates B cells and T cells to establish a lasting memory of the cancer, priming the immune system to hunt down cancer cells throughout the body. Imagine your immune system as a well-trained army and CPMV as the drill sergeant. It doesn't just wake up the troops, it reprograms them to recognize and attack cancer cells, even after the virus is gone. This means that CPMV could provide a long-lasting defense against cancer, unlike many current treatments. So what makes CPMV so special? The answer lies in its unique interaction with our immune system. While other plant viruses simply pass through our immune cells, CPMV activates a cascade of proteins known as interferons, which have powerful anti-cancer properties. This is a major breakthrough, especially when compared to other therapies that often come with hefty price tags and complex manufacturing processes. But here's a twist. Some scientists argue that CPMV's effectiveness might be due to its ability to mimic the body's own antiviral responses. This raises the question, are we simply hijacking the body's natural defenses? Or are we truly creating a new kind of cancer treatment? What do you think? Is CPMV the key to unlocking a new era of affordable cancer treatment? Or are we just scratching the surface of its potential? Share your thoughts in the comments below. Plant virus CPMV reprograms immune system to fight cancer. Breakthrough research. Cancer's unlikely foe. How a plant virus is revolutionizing immunotherapy. Imagine a world where a common plant virus holds the key to unlocking a powerful cancer treatment. Sounds like science fiction, right? But here's where it gets fascinating. Researchers at the University of California, San Diego have made a groundbreaking discovery that could change the face of cancer immunotherapy forever. The enigmatic cowpea mosaic virus, CPMV. Led by Dr. Nicole Steinmetz, a team of scientists has been investigating why CPMV, a virus that affects cowpeas, is remarkably effective at activating the immune system to attack cancer cells, while similar viruses fall short. Their findings, published in Cell Biomaterials, reveal the intricate mechanisms behind CPMV's ability to trigger both innate and adaptive immune responses, effectively reprogramming the immune system to recognize and destroy cancer cells. A closer look, what sets CPMV apart? In a side-by-side comparison with the closely related cowpea chlorotic mottle virus, CCMV, the research team, including graduate student Anthony Omelay, found that despite sharing structural similarities, CPMV's biological effects are uniquely potent. But here's the controversial part. CPMV achieves this without infecting human immune cells. Instead, it tricks them into an activated state, priming them to detect and eliminate cancerous cells. Unraveling the mechanism. So what's behind CPMV's remarkable ability? The data points to several key factors. Persistent RNA. CPMV's RNA lingers within immune cells, activating toll-like receptor 7, TLR7, a crucial receptor for antiviral and anti-tumor responses. Multivalent action. CPMV stimulates type 1, 2, and 3 interferons, known for their anti-cancer properties, and presents helper T-cell epitopes, adding an adjuvant effect. Tumor-agnostic efficacy. CPMV has shown promise in multiple mouse models and canines with cancer, inhibiting tumor-promoting signals and promoting long-term immune memory. The future of cancer immunotherapy. While these findings are preclinical and require further validation, they open up vast possibilities for leveraging pathogens from all life forms to develop innovative therapies. The big question, are we on the cusp of a revolution in cancer treatment? One that harnesses the power of unlikely heroes like CPMV? Join the conversation. Do you think plant viruses like CPMV could be the key to unlocking more effective cancer immunotherapies? What are the potential risks and benefits of using non-mammalian pathogens in human cancer treatment? Share your thoughts, and let's discuss the future of cancer research. Next steps? As the research team prepares for early-phase clinical trials, they are refining CPMV's formulation, dosage, and treatment protocols. The time for change is now. And with continued research, we may soon see the impact of CPMV in the fight against cancer. How a black-eyed pea virus trains your immune system to fight cancer. Imagine a world where a simple plant virus could hold the key to unlocking a powerful cancer treatment. It may sound like the stuff of science fiction, but researchers at the University of California, San Diego are making groundbreaking discoveries that could change the face of cancer immunotherapy forever. The cowpea mosaic virus, CPMV, a virus that typically infects black-eyed peas, has shown remarkable promise in activating the body's immune system to recognize and attack cancer cells. But here's where it gets really interesting. This virus is not only effective, but also potentially low-cost and easy to produce. In a study published in Cell Biomaterials, a team of chemical and nano-engineers took a closer look at how CPMV works its magic. They found that when injected directly into tumors, CPMV recruits immune cells like neutrophils, macrophages, and natural killer cells to destroy cancer cells. At the same time, it activates B cells and T cells to establish a long-lasting anti-tumor memory, which helps clear the targeted tumor and primes the immune system to hunt down metastatic tumors elsewhere in the body. But what makes CPMV so special? According to Nicole Steinmetz, the corresponding author of the study, it is fascinating that CPMV, but not other plant viruses, stimulates an anti-tumor response. The team's research gives us insight into how CPMV works so well. As Anthony Omole, the study's first author, explains, what we found most exciting is that although human immune cells are not infected by CPMV, they respond to it and are reprogrammed towards an activated state, which ultimately trains them to detect and eradicate cancerous cells. So, what's the secret to CPMV's success? To find out, the researchers performed a side-by-side comparison of CPMV with the cowpea chlorotic mottle virus, CCMV, a closely related plant virus that doesn't exhibit anti-tumor effects. They discovered that CPMV stimulates type 1, 2, and 3 interferons, proteins with well-known anti-cancer properties. On the other hand, CCMV stimulates a set of pro-inflammatory interleukins that don't translate to effective tumor clearance. Another key difference lies in how these viruses' RNAs are processed within mammalian cells. CPMV RNAs persist longer and get delivered to the endolysosome, where they activate toll-like receptor 7, TLR7, a critical component in priming antiviral and anti-tumor immune responses. But here's the best part. CPMV offers a unique advantage as a cost-effective immunotherapy. Unlike many other therapies that require complex and costly manufacturing, CPMV can be produced using molecular farming, which means it can be grown in plants using sunlight, soil, and water. The team is now working toward advancing CPMV to clinical trials, and their research has been supported by several prestigious organizations, including the National Institutes of Health and the American Cancer Society. As Steinmetz notes, The present study provides important insights into the mechanism of action of CPMV. We are diligently working toward the next steps to ensure that the most potent lead candidate is selected to achieve anti-tumor efficacy and safety. This is the time to move this work beyond the bench and toward clinical trials. But we have to ask, what do you think about the potential of plant viruses in cancer treatment? Do you think CPMV could be the key to unlocking a new era of immunotherapy? Share your thoughts in the comments below. And this is the part most people miss. The potential of CPMV to revolutionize cancer treatment is not just about the science. It's also about accessibility and affordability. With molecular farming, CPMV could be produced at a fraction of the cost of traditional therapies, making it a game changer for cancer patients around the world. But here's the question. Are we ready to embrace this new frontier in cancer treatment? Or are there still too many unknowns? The debate is ongoing, and we want to hear from you.